I'm delighted to welcome my longtime fellow group leader in the DRI, Valentina Escott-Price, also from Cardiff. Valentina is a professor of biostatistics in Cardiff and well known to the dementia community. She's going to talk to us about improving genetic predictors. Valentina, please take it over.
All right, so I will be talking about improving, as Paul said, prediction of Alzheimer's disease using UK Biobank. And I will tell you about this in two ways. One is about challenges to improve the prediction using UK Biobank data, and one is about advantages using UK Biobank data. So as we know about UK Biobank and the variables and the phenotyping which is available, I will be focusing today on two types of data: genome-wide genotyping and longitudinal follow-up for a range of health-related conditions. And why that? Because that's what my expertise lies in. So I will be talking, I'm best to talk about this, but I can talk about other topics too. So first of all, why Alzheimer's disease and what is specific about Alzheimer's disease? We know that about 60 to 80% been reported being attributed to genetics in terms of heritability of Alzheimer's disease. And we know that about 35% are risk factors which are environmental risk factors and which are potentially modifiable. Now, looking in details into the genetics, we know that 1% of cases with Alzheimer's disease attributed to very early highly penetrant mutations. Then we know that there is ApoE gene, which is high risk, common risk for Alzheimer's, latent set Alzheimer's disease, and it attributes for about 5 to 10% of heritability. We know that there are genome-wide significant loci, which identified now up to 92, and they contribute 8–10% now. And we know that there is a big chunk of other SNPs in the genome, which are not genome-wide significant, but still contributing to the heritability of Alzheimer's disease. And we will talk about how we can use all this information to predict genetically Alzheimer's disease. Interestingly, this is light green color. This is actually area which I'm mostly asked about for 15 years now. How do we use additional information? What are we missing? And missing heritability, you probably heard about this. And these are rare variants, copy number variants, and perhaps interactions, epistatic effects between SNPs, and copy variants which we don't know or don't know how best to detect yet. Another thing about Alzheimer's disease is that you have this ApoE. It's not only risk factor, it's also disease modifier factor. So E44 carriers have age at onset about 16 years earlier than those which are non-carriers, not E4 carriers. And also, once you get Alzheimer's disease, you have limited number of years to live, and therefore, when you look at the general population, the allele frequency of E4 decreases in the population. However, the prevalence of the disease increases in the population with age, we know that. And recently published paper in Sale Welsh database covering 80% of population of Wales, showing that this increase of prevalence of the disease in Welsh population is not only due to increased longevity in the population, but also people with dementia are living longer. So let's first focus on this dark green area, and that was quite well used for prediction of Alzheimer's disease genetically using so-called polygenic risk scores. When we include, that's the Manhattan plots showing association with with Alzheimer's disease. So we include all SNPs sitting above the line, these genome-wide significant ones, and also those SNPs sitting in this area, which perhaps not genome-wide significant, and we included into one number per person called polygenic risk scores. And what we observed, well, quite a while ago now, that we have good separation of the distributions of polygenic risk scores between controls, which are white here, and cases which are in gray. Now, how do we do it? How does it look in the UK Biobank? I looked at the number of publications trying to see if anyone tried to predict Alzheimer's disease using UK Biobank data, and I have not found too many. So I found papers which are related to using Alzheimer's disease genetic risk, predicting phenotypes which are related to Alzheimer's disease, like cognitive tests, imaging data, demographic data, loneliness data, etc., but not actually Alzheimer's disease. And what happens? Why? And that's the first challenge. So if you look at the general population, that's again picture from Welsh population, which is covering basically all sets of ages. The age at onset of Alzheimer's disease is about 80 years old in average. Of course, there is some early and later cases. And that is the distribution of age, the light color here, and age at onset of Alzheimer's disease in the UK Biobank. First of all, it's not so many, as we know, but also, you see, it's a majority of people have not reached the age of Alzheimer's disease as compared to general population. And that brings us, take into account ApoE, you know, my previous slide, early onset E44 carriers, that brings us to this investigation about heritability, which is captured by the data currently available in the UK Biobank. So look at this. So we capture about this UK Biobank genetic data, we capture about 30 to 40% heritability depending on the model. However, if we exclude the ApoE locus, you see the prediction drops quite a lot. Heritability, which we pick up drops quite a lot. And it's not because the models are wrong, it's simply because we have a lot of people in the UK Biobank who are still controls for this type of analysis. And that is a pathology confirmed data set. Similar if we include ApoE locus here, similar heritability estimates, and much higher heritability estimates if we exclude it, which means that we are picking up more in the later studies and with better clinically pathology confirmed cases and controls than in the UK Biobank at the moment. However, we looked also at heritability, which is picked up by particular biological processes, which are known to be or under investigation to be playing a role in development of Alzheimer's disease, and that's synaptic SNPs and microglial SNPs. And that's, you see, white circle is the SNPs in the whole genome, and that's the circles how many SNPs are involved in these pathways. So you see quite small number as compared to the whole genome. If you move to heritability, information which they pick up, you see it's quite a lot. So we are already can define or look at signal to noise ratio, and we can see that microglial and synaptic list of SNPs predicting, in fact, 80%, 82% of heritability captured by microglial list of SNPs and 58% captured by synaptic list of genes. That's already very good. We can estimate that, and that reassures us that we are doing the right stuff. But it's not only that. So what we also can do, we look at E44 carriers because we know that UK Biobank data is enriched for these people because they are earlier onset for Alzheimer's disease. And we can use that already to find something new, which we have not seen in case control studies or genome-wide association studies. So we included in this study only people who are E4 carriers, E44 carriers, actually, homozygous. And it was about 5,500 people. And we found, first time ever in my life, we found epistatic effects, and they seem to be real. Because that's a picture, the real epistatic effects, this was 10 to minus 5. We also found the gene, which was genome-wide significant in this particular small sample in E44 carriers, DUB1. And also we identified that this DUB1 gene lies in the pathway covering the DUB-reline pathway. And even more interesting, we found that risk alleles in that genes, they decrease age at onset, and protective allele decreases, as you expected. And recently, a new paper appeared in Nature Medicine talking about E33 homozygous person, but who is carrying presenilin mutation, which is highly penetrant. So, and they found that rare mutations in the same gene, reline gene, is associated with delayed age at onset. So we found something, first of all, in the UK Biobank, which has not been picked up by any genome-wide association studies, DUB1 and reline gene, and we found that these genes are biologically relevant, and also other people also seeing that. Furthermore, we have another fantastic now resource, it's clinical health records in the UK Biobank, and that's the first time I'm showing this result because the paper just submitted a couple of days ago. We looked at psychosis or psychiatric disorders and dementia. And what we found extremely interesting, that people here, you see all psychosis, mania, bipolar, depression, and anxiety. They have huge number, increased number of people diagnosed just prior to dementia, first onset, which is quite late if you look at general population and an onset for psychiatric diseases and disorders. We also found that more severe diseases like psychosis and bipolar have higher risk in the UK Biobank and in SAIL, we replicate that. And also we know that if you're hospitalized for this particular psychiatric illness, you even have higher rate of developing dementia. And this is excluding these guys. So here we have a new hypothesis which could be generated from analyzing UK Biobank data, looking at medical history, because we know that psychotic disorders, psychiatric disorders, they have their own genetics, their own pathways. And now we can look at people separately, those who had psychiatric disorders or not at all, and then develop dementia, perhaps it's one pathway, and those who have psychiatric disorders, it could be different group, target group, and genetic group. So, in conclusion, so message for you to take home. In Alzheimer's disease, ApoE locus impacts both survival, altering by age, and by increasing risk of other conditions, and frequency of E4 going down with age in the population. And despite being a young for dementia research cohort, the volume and the quality of the UK Biobank data allow us already to discover new genes, new biological mechanisms related to neurodegeneration, and also generate novel hypotheses to follow up. Genetic risk score, polygenic risk scores, there's still prediction accuracy about 70%, 70 up to 75. So not good enough yet for clinical implementation, but using all the information together, genetics, biomarkers, brain imaging, medical history is very important. It will provide us really much more accurate prediction of Alzheimer's disease. And we will move towards precision medicine, basically, looking at specific, because each person is individual, each person is different, and moving from prediction dementia in general, specific types of dementia, and even specific types of Alzheimer's disease. So I would like to thank my team in Cardiff and my colleagues from the Dementia Research Institute. And I'm happy to take questions at the end.
So, Valentina, there is particularly one question for DAB1, whether you've looked at this. And then I was going also to ask you, related to that, whether you have looked at interactions between the ApoE status and the PRS, or whether, the PRS is working differently in one subgroup versus the other.
So the answer is yes for both, to be simple. So the DAB1 gene is taken now forward for risk for biological analysis by my colleagues in the DRI at UCL. So they are looking into the biology of that, because I'm basically a drive lab person. So the second question, in terms of what did you ask? Was the second question?
About the ApoE status.
Yes, ApoE status, and yes. Yeah. So that is not as, yes, we looked at that. It's not statistically significant. However, we looked at that stratifying by age, and we know that PRS excluding ApoE is less predictive in younger people with younger age at onset, and more predictive in people with older age at onset, as I indicated and as I have shown. And what we looked at the interaction between ApoE4 and age, and that was significant in a number of samples, so we're writing up about this. But if you look just simply PRS, again, there are hundreds of ways of generating PRS, and we can discuss it forever. So basically, it depends how you generate it, so you can see different results and which sample you look at. Yes. So that was an answer.
Thank you very much.